Hi guys, I welcome you all to the next lecture in our anthropology series. So yesterday we had started the 9.4 unit, that is chromosomes and chromosomal aberrations in man. So I had given you a brief introduction of how the chromosomes look, so I'll just revise that also with you. So there are basically four, that is metacentric, this one, the third one, then submetacentric, acrocentric and the telocentric. We had studied about this. This is called the centromere, you can see, right? It is called the centromere, and these are called the short arm and these are called the long arms, and you can say P arm and Q arm also, right? So this is how the chromosomes look like, and this is going to be helpful in when we are going to study the chromosomal aberrations. So we had divided the chromosomal mutations under two parts. One was the number, which was called the heteroploidy, and the other was the chromosomal aberrations, wherein we had to study the structural part, right? So structure we are going to see today. In this we had already studied monoploidy, polyploidy and like that, right? And euploidy, aneuploidy, right? So all of that we had already studied. I'm not making it today again because it is not the topic of discussion. Today we are going to do the chromosomal aberrations, that is how structural changes or how there are changes in the mutations that take place in the structural aspect of the chromosome. So now you must be intrigued ki what happens in this. So let's see, right? Okay. So chromosomal aberration is where the structure of the chromosome is going to change, and thereby there is certain kind of a mutation that occurs, right? So it could be deletion, ठीक है? It could be duplication, ठीक है? It could be translocation, ठीक है? These are major headings. What can happen we are going to see. Don't worry about that. And there could be, you can say, inversion, ठीक है? There could be ring, isochromere. So these are certain headings that we are broadly going to study. Okay, so let's look at the first one, that is deletion. Okay, so yeah, just look at these pictures. Herein I have shown you deletion, this is inversion, this is isochromere, ring, insertion, translocation, etc. Now we are going to draw each one of them and understand. Okay, so now the first one I said, because I'm understanding you will not be able to understand it with the picture, so I will draw it for you also. So in deletion, as the word suggests, delete. So that means there is loss of a part or segment of a chromosome that leads to loss of genes. Now just understand what is happening. We are saying, let me just draw it for you. This is a chromosome, fine. This is a chromosome. So obviously it has certain genes which has certain encoded information, or the genetic material, you can say it has the genetic material. Okay, what is genetic material? The DNA, all of that, right? So if any part of or any part or whole segment, now segment क्या हो गया? I may call this as a segment of the chromosome. Okay, this is a whole segment, right? So if any part or segment of a chromosome is missing, so we can say it means that there is some deletion that has happened. So if I draw a chromosome like this, so it should have been like this, right? It should have been like this, but now what happens is it becomes only this much remains, and rest part cuts out or gets deleted. This part जो यहाँ पे ये part था, I am drawing this separately. So now this part has got deleted. So now obviously certain diseases are going to occur. So examples, you can say that there's a disease called Philadelphia 22. So it occurs, there is deletion that occurs in the chromosome number 22, so it is called Philadelphia, right? सो डिलीशन में आप एग्जाम्पल दे सकते हो, और यू कैन गिव एन एग्जाम्पल ऑफ क्राइडू चैट डिजीज. This is a disease in the children, cry-do chat, because in this the cry of the baby becomes like a cat. Okay, so the cry of the baby sounds like the cat, so that is why it is called cry-do chat. And herein the deletion happens on the short arm. That means obviously short arm to ye ho gayi, right? So in deletions happen on the short arm ऑफ क्रोमोसोम नंबर 5. ठीक है, सो हमने डिलीशन देख लिया. व्हाट इज डिलीशन? वेयर इन अ सेगमेंट और अ पार्ट, either the whole segment or a part of sub part of a chromosome could be missing. Now don't confuse, you can either draw it the chromosome like this or you can draw the chromosome like this, it's okay. ठीक है, we are just trying to understand what is happening. So deletion में क्या हो रहा है? इधर मैंने आपको I tried to show you a diagram wherein मैंने what I did, I deleted this segment and I showed it separately. That means this is now the it should have been like this, it should have been like this, but it is only this much. Okay, so herein ये part missing हो गया, right? So in this I told you there are two diseases which happen because of this, wherein sometimes if the short arm, in the short arm of chromosome number 5, if it is missing, so it causes cry-do chat disease, and there is a Philadelphia disease also that happens because of the deletion. So let's go back and let's understand. And deletion, now you will understand the picture. That means it should have been this much, right? It should have been this much, but this much part is missing. ठीक है? Okay, so deletion you understood. What is deletion? Okay, so इसमें we have written also one more example. Wolf, I don't know now how to pronounce this, Hirschhorn syndrome. There is partial deletion in the short arm of chromosome number 4. So you can see there are a lot of diseases that happen due to such kind of chromosomal aberration. And you saw now why this is structural, because the structure has changed. The normal structure of the chromosome looks different in all the cases that we are seeing, right? So there is structural difference. Okay, now let's go to duplication. Okay, let's see how duplication happens. Okay, okay, so now again पहले मैं normal, first I'll draw the normal one and then the not so normal one. Okay, so let us say that this was the normal chromosome and there is this segment which I am just marking. So this is normal, and in the duplication what happens is this segment which I have like highlighted, you can say, gets duplicated. So it normal should have been like this, but now it has become like this. That means this part has duplicated itself, right? So I think there is no doubt about this. What is this concept? Whole concept. And in this there is a disease that is called Charcot-Marie-Tooth disease. Okay, so this happens due to sometimes there is duplication that occurs, and this is a very common example of duplication. Okay guys, so this happens in the chromosome number 17. Okay, now you need not to remember which chromosome it happens and what diseases. This is just for your understanding. I am giving you that this disease happens and this and all that. You just need to have a understanding of it. So in here also we must have a picture of duplication. I think there is no picture of yes, you can okay, there is no picture of duplication. Okay, next is see, I have written also Charcot-Marie-Tooth disease, chromosome number 17 में होता है, right? After that, translocation, when a part of chromosome gets transferred to another. So in this translocation what happens, there is a portion that gets translocated to some other. Okay, so for example you can see this is translocation. There were two chromosomes, but some material of this transferred to this and some material of this transferred to this. So this is also called as a balanced translocation. okay, because you can see there is a balanced translocation. Sometimes it might not be balanced translocation also. Now I will show you only. Who is going to show you? I am only going to show you these pictures also. So what is balanced translocation? Let me first draw two. So this is, for suppose there were these two normal chromosomes. Okay, these are normal. Now translocated ones I'll just show you. Translocation. Okay, this is balanced translocation. Otherwise it should have been looking somewhat like this, but now some of the genetic material of blue has gone to yellow, and some of the material of yellow has gone to blue. Okay, so this is one example of and you can call this as balanced translocation also, but many a times it is not balanced translocation. That means you can say that so herein what has happened, there is deletion in this one and translocation in this one, so it is not balanced, right? Balanced translocation, and in the second case there is deletion from the one and translocation in the other. So ye isme there can be cases like this also. So one of the translocation, or isko you can also call it as reciprocal, where both have, you know, like reciprocated to each other. So you can call it reciprocal also. And this example of translocation you can say is Robertsonian disease, Robertsonian syndrome, you can just call it. Okay, it occurs in, you can say, 13, 14, 15, 21 and 22. So in these translocation, if these number of the chromosome number 13, 14, 15, 21, 22, if it happens, it is called the Robertsonian syndrome. Okay, so you understand, right? Okay, so next is, okay, so I've written it also here, Robertsonian, and it is a part of translocation. Now comes inversion. Now what happens in inversion? The part of or portion of a chromosome breaks, turns itself upside down and gets reattached. So genetic material gets inverted. For example, now this might sound very tough to you, what is happening. So you can just see if there is a picture. Yes, I have put a picture here. This shows inversion. So but I'll make it a lot more clearer picture for you. Okay, so basically normally what it should have been like this. So let us differentiate now, so otherwise you will not be able to understand. This is a normal chromosome. Let's take this as a normal one. Now what happens in inversion? A part breaks, that means you say this one has broken itself, okay, turns itself upside down and gets reattached. So in this case you would say that it has broken and reinverted itself, so it becomes like somewhat like this. So okay, I'm sorry, this would happen in the green color, they should be in the green, and now that part has now reattached itself here. So you understand what has happened? The deletion happened in one part, it got inverted and reattached itself. So the genetic material also got inverted. Okay, it was somewhat like this, now it has become somewhat like this. So genetic material also inverted. So this is the example of inversion. I hope you understand what inversion is, right? So now let's go. This is inversion. Okay. Okay. Next comes ring. Now इसमें क्या होता है? In ring, forms ring or circle can happen with or without the loss of genetic material. So ring, it's very like simple. You can just see the chromosome should have been like this, but what it becomes, it forms a circle. So from here you can say it reattaches like this. So it should have the arms should have been straight, but they become circular and they reattach themselves like that, right? So this becomes a ring. Okay. And then comes isochromomere. Now isochromomere, in what happens? Mirror image of chromosome including the centromere. Now what happens, you just understand. There is deletion of one arm, okay? Then there is duplication of the other arm, and then it reattaches itself. I'm sure now you have not understood this. I'll just tell you what happens. Don't worry. Just look at this. Let me draw a normal one first. Okay, let me draw it with two colors so that you are able to understand. Okay, this is normal. Now what happens, let me say that green part now gets altogether delete हो जाता है, ठीक है? So now what happens, green one gets deleted and the yellow one duplicates itself and gets reattached, ठीक है? So there is loss of one arm, ठीक है, which is the green one, and there is duplication of the other, and then it gets inverted, then it gets attached in the inverted manner. That means yellow ऐसे था, it should have been like this, but now it has duplicated and put itself like this also, right? So this happens in isochromomere. So what do you understand by isochromomere? It is different than inversion because it is, you can say it is a mixture of deletion plus inversion, right? So what's happening? Firstly, one arm deletes itself. The second arm duplicates itself. So it's duplication also, right? So the second arm duplicates itself and then inverts itself, right? So what is happening? Deletion, duplication, inversion. So this is isochromomere. So you understand what is isochromomere, right? So I hope you have understood these genetic disorders, right guys? So if I have to tell you by just showing whatever we have done the structural changes, okay? We have done the numerical changes that happens, and then we are going to cover in this chapter some other kind of genetic disorders, because basically why are these genetic disorders happening? These are certain mutations, it could be mutations, right? Then there is genetic disorders which can happen in your autosomal chromosomes, and they could also happen in your sex chromosomes, or mostly it is the X linked, because very few genetic disorders are in the Y chromosome. I told you, because X encodes more genes, more genetic material is there, that is why X linked diseases are more, right? So some of the examples of X linked, you can say hemophilia, and then there is, you can say, red green blindness. We are going to do it, we are going to study it, don't worry. Then there are autosomal mein there are autosomal dominant and there are autosomal recessive, right? So in autosomal dominant you can say there is Huntington disease, okay? Then in recessive you can say there is sickle cell, right? Sickle cell is there, okay? Then we are going to study certain other kind of genetic disorders also, which are based on the pattern. For example, on the non-disjunction of the chromosomes. Okay, many times during the meiosis, the chromosomes do not detach properly. So that is why the numerical are there, and there are so many others. In pattern में, नॉन डिसजंक्शन में यू कैन से डाउन सिंड्रोम, टर्नर सिंड्रोम, जिसमें ट्राइजोमी वाले मोस्टली इसमें आते हैं, बिकॉज दे आर नॉट एबल टू डिटैच, ठीक है? सो नॉन डिसजंक्शन में आते हैं. ऑल दीज आर द टॉपिक्स फॉर टुमारो, डू नॉट वरी, आई जस्ट गिवन यू अंडरस्टैंडिंग. हमने बेसिकली देख लिया क्रोमोसोमल एबरेशन ईदर न्यूमेरिकल होगा और इट विल हैपन इन द स्ट्रक्चर. नाउ दीज आर all the you can say different typology. The concept is only number or structure. We have now put them under different classification, so the classification would vary, but the diseases are the same. And then we are going to study certain specific disorders that are mentioned in the syllabus. So I hope guys you have liked the lecture for the day. If you did, guys, please like, share and subscribe. Thank you so much.